Hey guys, I hope you're doing well. I hope you're hanging in there. I know this is some strange times we're living in, and certainly not the easiest environment in which to learn, but hang in there. We'll get through it. Today's lesson covers pages 286 to 288 in your textbook, so make sure that you've read those pages before you view this lecture. And the topic today specifically covers RNA processing. So we're going to be talking about the events which occur in a eukaryotic cell, animal cell or plant cell or fungus cell, after transcription, but before the RNA actually leaves the nucleus. So in a prokaryotic cell, bacteria, the process of translation just happens immediately after transcription has concluded. But there are some extra steps that take place in a eukaryotic cell right after transcription. That's what we're going to talk about today. Okay, so specifically, what processes occur after transcription but before translation in a eukaryotic cell? Specifically, there's three things that happen. There are two things that are added to the messenger RNA after transcription, and then there is some editing, some splicing or removal of things from the RNA. So it's kind of strange. Two things are added to the RNA, and then some things are removed from the RNA. So we're going to talk about the 5' cap, what that is, why it is added to the messenger RNA, the poly A, that is many adenines, poly A tail. Those are the two things that are added. And then we're going to talk about this process known as RNA splicing, in which introns, I'll explain that term, are removed. And in some cases, alternative versions of the RNA are created through an editing process, which allows for a single gene to code for different protein products. So all of these things that we're talking about today that happen after transcription but before translation are simply collectively referred to as RNA processing. So this happens post-transcription, right after transcription. Okay, so here's a look at what we're talking about. The pink strip that you see here represents a single strand of messenger RNA that's just been made through the process of transcription. So this would be your A's and your G's and your C's and your U's. And then the yellow represents the two things that are added. You can see we're adding something to either end of this RNA. And so at this point, the messenger RNA that you see, we would refer to this as pre-mRNA. It's not mature yet. It's not ready for release. It's not ready to leave the nucleus. But part of getting it ready to leave the nucleus is to add what's called a 5' cap. And the reason it's called that is because it's added to the 5' end of the RNA. What is the 5' cap? It is a guanine, a single guanine, that is bonded to 3 phosphate groups. So it's GTP, guanine triphosphate, similar to ATP, but ATP would be an adenine and 3 phosphates. This is a guanine and 3 phosphates. So the guanine triphosphate is called the 5' cap, and then on the other end, on the 3' end, many adenines, extra adenines are tagged on to this end. So it's poly A, many A's. It's called the poly A tail. So here's some more information about that. The 5' cap, as I said, is guanine triphosphate. It's added to the 5' end. And there's probably at least 2 reasons why this happens in eukaryotic cells. It protects the messenger RNA from degradation. Now what that means is once this molecule leaves the protected sanctuary of the nucleus, when this gets out into the cytoplasm, there are all kinds of enzymes that will eventually basically eat this molecule or slowly degrade or break down this molecule. So if you didn't have the 5' cap, if the very end of this molecule was the important letters that are part of the actual code, then that code would immediately begin being destroyed by these enzymes, these hydrolytic enzymes that are in the cytoplasm. And messenger RNA is meant to be a temporary molecule. It's not supposed to last forever, but it does need to last long enough for translation to occur. It needs to survive long enough for at least one ribosome to attach to it and build a protein from these instructions. So by adding this 5' cap and also by adding the poly A tail, you're putting some buffer on either end. So if an enzyme starts eating away at this molecule, it's going to be eating away at the 5' cap, or it's going to be eating away at the poly A tail, instead of immediately destroying this precious code that's in here. So it's a protective cap and a protective tail that's placed on either end. So that's one reason for the 5' cap. It's also very possible, some of this we don't know for sure, but it's thought that the 5' cap helps the ribosome attach properly. Because when it's time for the ribosome to attach and begin reading these codons, you don't want the ribosome to start at the 3' end, because then it would be reading the message backwards. It would be like you trying to read a sentence from right to left, and it wouldn't make any sense. It would just be a garbled message. So we want the ribosome to attach to the 5' end and then read in a left to right direction, or a 5' to 3' direction. And so the 5' cap is sort of like a signal that says, hey, ribosome, you need to attach on this end. Don't attach down here where the poly A tail is. The poly A tail is just that. It's anywhere from 50 to 250 adenines. A, a whole bunch of A's added to the 3' end. This probably also slows down the degradation or the destruction of the molecule by enzymes, as we described earlier. And it probably also helps the ribosome attach. In other words, it's think of that poly A tail as a signal to the ribosome that says, hey, dummy, don't attach to this end. This is the wrong end. You need to attach to the guanine triphosphate. You need to attach to the 5' end so that the code will be read correctly. So the 5' cap says, hey, ribosome, attach here, and the poly A tail says, hey, ribosome, don't attach here. This is the wrong end. It is also thought that the poly A tail somehow helps the messenger RNA find its way out of the nucleus. Remember, the nucleus, I don't have a good picture right here to show this, but page 297 in your textbook shows a strip of messenger RNA in the process of leaving the nucleus. And you can see that it literally has to thread its way through pores, the little tiny openings in the nuclear envelope. It's almost like threading a needle, putting a tiny filament through a very tiny opening. And it's thought that the poly A tail and maybe even the 5' cap somehow help that process, help facilitate that process of the messenger RNA exiting the nucleus. There's a lot we don't know about this. There's a lot that people are still trying to figure out. But those are the two things that are added to RNA before it leaves the nucleus: 5' cap and the poly A tail. Okay, and then remember there's a third event that happens. The 5' cap is added, the poly A tail is added, but there's also some things that are removed. So before we describe that, let's talk about exons, not the gas company, exons and introns. Whenever we look at a nucleotide message, A's and G's and C's and U's, if we're talking about RNA, but even if it was DNA, A's and G's and C's and T's, it's important for you to know that in a eukaryotic gene, not all of those letters actually are part of the genetic message. There are little segments within the code that don't code for anything. They're not part of the gene. Now, this is not true in bacteria. In prokaryotic genes, there are no introns. The entire code is part of the code in a bacterial cell. But in a eukaryotic cell, animal cells, plant cells, fungus cells, protist cells, and I just learned this recently, even some viruses, there are introns. So what does that mean? Well, the dark pink that you see, those are the exons. An exon is a segment of, it could be RNA or DNA, but in this case we're talking about RNA because this is the pre-mRNA. This is what was just made in transcription. So an exon is a segment of that RNA that will exit. That's where the name exon comes from. It's going to exit the nucleus, and it actually will be expressed. So that's how you remember it. Exons are expressed. Exons actually do contain codons, and they are coding for amino acids so that the protein can be built. But introns are just in the way. They are intervening pieces of RNA, or again, could be DNA if we were talking about the actual DNA gene. But since here we're talking about RNA, this is after transcription, an intron is a segment of intervening RNA that's interspersed within the strand, but it will not be expressed. It is simply in the way. Exons are expressed, introns are just in the way. So here's my metaphor for this. I like metaphors, and I love Star Wars. So I'm going to use the example of Star Wars here. Okay, in 1977, when George Lucas was filming the original Star Wars, what came to be known as Episode 4, A New Hope, think of the pre-mRNA as the raw film footage. Back then they actually shot movies on actual film. It wasn't digital, of course. And so the dark pink and the light pink, this represents the raw film footage that they shot as they were making that movie. Hundreds of hours of shots, And when you make a movie, there's going to be scenes that you have to shoot more than once. There's going to be bloopers. There's going to be times when the actors forget their lines, or they mess up, or the special effect doesn't happen just exactly right, or the stunt messes up. And so there's going to be scenes that you're going to cut later. But initially, it's all on one continuous piece of film, at least back in the pre-digital days. So you're going to have segments that you are going to use, that you are going to keep in the final film, but you're going to have segments, maybe whole scenes of the movie that you're eventually going to cut. And a famous example of this in that Star Wars movie in 1977, there was a shot that was originally written in the script where Han Solo goes to meet with Jabba the Hutt to explain why he did not deliver some cargo that he was supposed to deliver for Jabba, and he's sort of pleading his case and saying, please don't kill me, please don't, put a hit on me, because I'll make it good. Well, that scene was cut out. They decided the movie was running too long. They didn't have enough special effect capability to make Jabba look real. So what they did was they cut that scene out. And back in those days, you would literally make a cut on the film here and here. You would make a cut. And that piece of film, that whole scene that you wanted to edit out, would be thrown on the floor. They called it throwing it on the cutting room floor. And somebody would sweep it up at the end of the day after the editing and throw it in the trash. And so they would take the scene on either side of that and literally glue the film together or kind of melt it together. It's called splicing. And so we call this process RNA splicing. It's totally just a stolen term from the old days of filmmaking. So we're going to cut out the scene that we don't want in the final film. We're going to cut out the bloopers and throw them away. And what we end up with in the movie theater is the polished, finished product with all of the introns cut out and the exons all joined together in one continuous strip. So this would be called the mature messenger RNA. It's ready to be released. It has its 5' cap, it has its poly A tail, and all of the introns have been spliced out. Okay, so part of RNA processing is what I just described. It's RNA splicing. The introns have to be cut out. Introns are non-coding nucleotide sequences that are just in there. They're in the way. They are cut out. And the exons, which are the coding regions, the dark pink, those are spliced together to make a finished product of A's and U's and G's and C's that actually do code for the protein that we're trying to build. So remember I mentioned that the way this happens and the way it happens in filmmaking is that there's literally a cut that's made on either side of the piece we want to get rid of. Well, who makes that cut? Well, in Hollywood it would be a film editor, but in your cells it's a mechanism called, and I'm not making this up, it's called the spliceosome. What is a spliceosome? Well, it's a whole bunch of proteins. And this is my favorite acronym in all of biology, SNRPs. I'm not making that up. SNRNP stands for small, nuclear, because they're small, they're in the nucleus, ribonucleoproteins. These are little pieces of RNA that are combined with proteins to make editing mechanisms, SNRPs. Okay, the little pieces of RNA put together with proteins, and all of this joins together to make this huge structure inside the cell that attaches to the RNA, cuts out the introns, and splices together the exons. So initially, we would call this pre-mRNA before it's edited, when it still has its introns, and then after the introns have been spliced out, we call it mature messenger RNA. So that's the mature messenger RNA right there. This is what we're actually going to release to the cytoplasm so that the ribosomes can attach to it. Okay, here's a closer look at what a spliceosome is and how it works. So here is a SNRP, a small nuclear ribonuclear protein, and you can see the pink strip represents a piece of RNA, and then the sort of purple oval is a protein. So it's a combination of a protein and RNA, and then there are other proteins that come in, and all of this combines to make this big mechanism. It's almost like one big functional enzyme called the spliceosome. The spliceosome attaches to the area that we want to edit. One of the SNRPs attaches to the place where we're going to make our first cut. Let me see if I can switch to my pen. So we're going to make a cut right here. And then another SNRP attaches to the other end of the intron so we can make a cut right here. Ooh, I'm getting kind of messed up here. And those two cuts allow the intron to sort of be bent out and removed, and excised means it's literally discarded. It's cut out and thrown away, and eventually it's degraded. Sometimes there actually are functions that these introns will carry out. We won't get into that part of it, but usually they're just degraded and broken down. Now the dark pink is the exon, remember, so we're going to keep that. And here's another exon that we want to keep. So after those cuts are made and after the intron is cut out, the exons are going to be sort of glued together. They're going to be bonded together covalently by the spliceosome. And so now we used to have two exons with an intron in between them. Now we've just got the two exons that are bonded together. Okay, so this is a good time to mention today's code word, and you know what to do with the code word. Today's code word is SNRP, and even though it's pronounced SNRP, it's actually spelled SNRNP. That's today's code word. Okay, now I haven't even told you the coolest thing about RNA splicing, and this again is very similar to what happens with film editing. It turns out, we've learned this fairly recently, this is a fairly recent discovery, I'd say the last 15 years or so scientists have started learning about this, is that when you edit RNA, it's not always edited the same way. So the same gene, after transcription, can be edited different ways in different cells. Now that's pretty cool. What that means is you might have a gene that codes for one protein in, say, a nerve cell, and that same gene might code for a totally different protein in a blood cell or a muscle cell. How is that possible? Well, let me show you. It's called alternative RNA splicing. It means different final messenger RNAs can be made from one primary transcript. In other words, different mature RNAs can be made from one pre-mRNA. When the exons are spliced together, there are some things that can vary. So which exons are included, because they don't always have to all be included, and which exons are left out. I'll show you what that means in just a minute. That can vary from cell to cell. And it means that this is really the most important thing to know. Alternative RNA splicing means that more than one protein is possible from a single gene in a eukaryotic cell. This would not be something that would happen in a prokaryotic cell because they don't have introns. The overall concept and even the terminology is very similar to film editing. I've already kind of alluded to that. So to stick with my Star Wars example, again, when the original Star Wars was shot in it was probably shot in 1976. It was released in 77. I'm just making this number up, but let's say they shot 120 hours of raw footage, probably more than that. Most of that film footage got edited out, right? Because they had to shoot the scenes over and over again, and they had to find just the perfect scene where the actors got their lines right and everything happened just the way George Lucas wanted. So most of the film was edited out and fell on the cutting room floor. Those were the introns, the scenes that were cut out. And that included some scenes that were completely cut out that never made it into the final film. I mentioned this scene between Han Solo and Jabba the Hutt, which was originally shot. They had a human actor playing Jabba, and they were going to figure out how to superimpose a special effect on top of him to make Jabba. They weren't even sure what Jabba would look like in 1977. But anyhow, this was a scene where Han Solo was going to plead his case and beg for his life. And when I went to the movies in 1977, I was only 4 years old, but I remember going and watching this movie. That scene was not in there. It was edited out. However, if you were to go to Disney Plus or purchase a DVD of Star Wars: A New Hope and watch it today, you will find that scene is included in the movie. So it's a different edit of the movie. Today, the movie has been re-released with that scene added back in, with all the special effects in place. So it's possible to take the same raw footage and edit it in two different ways to get two different final products. There are several differences actually in the most recent released version of A New Hope compared to what I saw in the theaters in 1977. There's several scenes that have been altered. So that's what alternative RNA splicing is. So here's a gene. This is a single gene. Like all eukaryotic genes, it has exons and introns, exons and introns. You can see there's 6 exons. The dark blue represents the introns that we're eventually going to splice out. This is the DNA. So after transcription, this would be the messenger RNA, the single-stranded messenger RNA. It also has all 6 of the exons, and it has all of the introns. Now, in RNA processing, we know the introns are going to be cut out. Like we talked about earlier, there's going to be a cut made here, there's going to be a cut made here, and we're going to get rid of this intron, and we're going to splice together these 2 exons. When that editing takes place, one way to edit this would be to remove all of the introns and put all of the exons together, or at least most of the exons together. You can see they actually left out exon number 3. So this time we spliced together exons 1, 2, 4, 5, and 6. So this would be a final edit that could be released from the cell. The ribosome would read this and build this protein. And this protein has some function that it has to do inside the cell. But in a different cell in your body, that same gene, after transcription, could be edited a different way. What if we left out exon number 2 and then put exon number 3 back in, and we edited it this way? Well, it's a different message. It's like Star Wars with the Jabba the Hutt scene versus Star Wars without the Jabba the Hutt scene. This is going to code for a different protein. It's not the same protein. It's not going to have the same function. It's going to do a different job in the cell. And it's possible that that same piece of messenger RNA could be edited yet a third way. Maybe 4 or 5 different ways you could edit this. Sometimes when, with a movie, you have like the theater version, and then you have like the director's cut, and then you might have the 25th anniversary special edition with, different scenes that are added back in. You get the idea. But what does this mean? It means that many different proteins, we didn't used to think this was possible. Now we know many different proteins can be made from one single gene, because of alternative splicing of messenger RNA. You don't always have to include all of the exons when you splice them together, and by editing this different ways, multiple different proteins can be made. So if we go back to the idea of the central dogma, you remember this from a previous lesson, the old idea since the 1950s was that every single gene codes for one piece of messenger RNA, which codes for one polypeptide. So in 2003, when the Human Genome Project was completed, and we figured out, okay, humans have 20,000 genes, well, that means if you follow the central dogma, if we have 20,000 of these, then that means we can make 20,000 different messenger RNAs, and that means we can make 20,000 different proteins. But that's not true. We know that human cells can make well over 200,000 different proteins. How can that be? If we only have 20,000 genes, and that means we can only make 20,000 different pre-mRNAs, how the heck do we make so many different kinds of proteins? Well, now you know. It's because after transcription, when we get to this point right here, those pre-mRNAs can be edited multiple different ways. And there's probably more to it. There's probably a lot going on here that we don't know about. But part of the answer to this is that alternative RNA splicing allows us the possibility as eukaryotic organisms to make many, many different proteins from each one of our genes. So we can make way more different proteins than we have genes. Alternative RNA splicing is important indeed. Okay, our next lesson is going to focus on translation. So we went through the process of transcription, and then we just talked about what happens after transcription. And next we're going to talk about the process of translation. Again, you learned about this last semester, so we're just going to go into more detail. These are the pages that you need to read. And just to make sure you understand, the idea here is for you to read the material first. That's really important, so you kind of have a baseline of what we're going to talk about. Then watch the lecture. Please take notes, just like you would in the classroom. I know this is tedious. Okay, I get it. But the nice thing is you can watch it as many times as you need to. You can keep backing it up if you say, what the heck did he just say? Watch it, take notes, then study those notes. Go back and watch it again if you need to, and then take the quiz. Those 10-question quizzes are closed note. They are not meant to be open note. You don't have time to look up the answers in your notes. So I just want to make sure that I'm clear on that. Guys, I miss you. I hope to see you soon. It might not be anytime real soon, but y'all take care and be safe.